My name is Nataly Delgado, and I'm an undergraduate student majoring in biological sciences at the University of Texas at El Paso, otherwise known as UTEP. I have been a member of Dr. Kyung-An Han's Neuromodulation Disorders Lab since the beginning of summer of 2017. The way that I got into research was through a collaborative program between El Paso Community College, otherwise known as EPCC, and UTEP. It is called Bridges to the Baccalaureate Summer Research Program. Even after knowing very little about research, I decided to try it out because I did not know if I was going to be given another chance to pursue research. I expected it to be very difficult to find opportunities, but on the contrary, there are many programs at UTEP that encourage students to get a head start into research. This program allows transfer students from EPCC to participate in biological research at UTEP and encourage the building of connections between UTEP staff and the student before transferring. Before being selected into the program, I was able to meet with different professors and discuss their work. Through this system, I met my current mentor, Dr. Han. She has guided me through the participation of various programs such as Summer Undergraduate Research Program Assistantship, also known as SURPASS, as well as my current program called the Research Initiatives for Scientific Enhancement, or RISE. Throughout my years in the lab, I have been collaborating in different projects around my lab, but my main project focuses on finding the role of the dopamine D2 receptor in ethanol-induced behavioral disinhibition and sensitization using the Drosophila melanogaster as our animal model, or better known yet as the fruit fly. Alcohol is a psychoactive drug that has been used and abused by many for a long time and can be highly addictive. Its effects are known to cause disinhibited behaviors such as euphoria, risky sexual behaviors, and locomotor impairment. Yet its neural mechanism remains unknown, which is why the focus of this study is to clarify the neural pathway of ethanol-induced behaviors such as disinhibition and sensitization. In the wild, male flies rarely court other males, but previous research in my lab has found that under chronic ethanol exposure, there is an increase in the amount of inter-male courtship. It is known that dopamine is a necessary neuromodulator and neurotransmitter responsible for behaviors associated with alcohol intake and addiction. The focus of my project is to determine if the dopamine D2 receptor is necessary for this ethanol-induced behavior and its functional site. In our study, we chronically exposed wild-type Cantonese male flies and D2 receptor mutant flies to ethanol and monitored their behavior. My findings suggest that this dopamine D2 receptor is necessary for ethanol-induced inter-male courtship disinhibition. Currently, I am working on finding which major D2-like receptor isoform or isoforms are necessary for this behavior and mapping the tissue-specific location in which it is important, as well as interaction with other previously characterized dopamine receptors. Being a part of this lab has allowed me to learn various methods such as immunohistochemical analysis, which allows us to visualize proteins in the brain with the help of antibodies. I have also learned how to use the use of genetic tools to manipulate animal models to knock down or induce genes such as the Gal4-UAS system, RNA interference, gene switch, and the target system. The Gal4-UAS system is a binary system that allows us to express a gene of our interest and express it in a tissue-specific manner. Also, different methods to measure fly behaviors and ethanol delivery systems, which is the focus of my project. I have grown to really enjoy the research I have been investing on. It has allowed me to understand most aspects of how a career in academia will suit my personal interests, as it is my goal to pursue a path in neuroscience research focusing on neural mechanisms affecting addiction.